Okay, so today we are going to discuss a very important topic in pharmacology, that is smooth muscle. You know, now a very important and intrinsic topic is smooth muscle. So many drugs, antispasmodic drugs are associated with this. Therefore, I thought that I should take this class first before going into the smooth muscle, pharmacology or antispasmodics. Okay. Now, a very important point we need to understand is this: that smooth muscle. If you want to learn smooth muscle, you will have to remember these 2 points first. What are those 2 points? That is, smooth muscles. Smooth muscles are the most abundant. They are the most abundant muscle in human body. Clear? They are the most abundant muscle in human body. Clear? Now, second point you need to understand or you need to remember at first is this, that second point you need to understand is this, that smooth muscles are non-striated muscles. Okay, they are non-striated muscles. They are non-striated muscles. What happened to the markers? It's all bad here, Nothing is working. Okay, let us try. Yes, this is working, thank God. Striated muscle. Now, we will talk about everything, why it is non-striated and all these things. Now, the first two things you need to understand right now is this, that whenever we are talking about smooth muscle, the first thing that should come into your mind is that it is the most abundant muscle in human body, or rather in your body. Okay, if you are human, then. And second point is this, you are laughing, that means you have a doubt. Great. Now, listen. And the second point is this, that they are non-striated muscle. Clear? These 2 points you need to remember first when you are, starting this particular topic. Clear? Okay. Now, we will actually, discuss smooth muscle. We will discuss smooth muscles with respect to three main, we will discuss smooth muscle with respect to three main, points. That is, the first point is its structure. Second point is contraction. The third point we need to understand is this, its types. Clear? Its types. So we will actually learn smooth muscle on the basis of three main topics. That is, the structure of smooth muscle, the contraction and contractile properties of smooth muscle, then its types. Clear? Now, a very important point here is this, that we will be learning the structure first. Okay? Clear? We will be starting with the structure of smooth muscle. Now, listen. Do you know that smooth muscles, not all smooth muscles, but many smooth muscles have a nickname that is called, they are visceral muscles. That means, what do you mean by viscera? What do you mean by this term viscera? Viscera. You know what is meant by this term viscera? Viscera means internal organs. That means, smooth muscles form the bulk of, our what? Internal organs. Like best example of your viscera is what? Which is filled with smooth muscle? Stomach. Clear? Now, okay. So now we will be talking about the structure of smooth muscle. We will be talking about structure of smooth muscles. Okay. Clear? We will be talking about the structure of smooth muscles. Okay. Now, very important point we need to understand is this. Suppose this is your stomach. Okay. Suppose this is your stomach. Okay. Now, if we cut a three-dimensional piece from this area of smooth muscle, what we will see is this. Okay. What we will see is that this is the three-dimensional structure. Okay. And suppose these are the muscles. These are the muscles. Now what we will do is we will actually take out one of the muscle, one of the smooth muscle, and we'll draw it here, but we need larger spaces. Okay, we need larger spaces. Okay, now listen with all your attention. Suppose this is your smooth muscle. This is one smooth muscle fiber. Okay, this is one smooth muscle fiber. Now, a very important point we need to understand is this: unlike skeletal muscle and cardiac muscle, the nucleus of smooth muscle is actually in the center. Clear? In the center. Clear? In the center. Now, so we need to understand these basics first. Now, we will actually start with the membrane of smooth muscle. Okay, we will actually start with the membranous structure of smooth muscles. Now listen with all your attention. Now suppose this is a smooth muscle, and if we start with the membrane of smooth muscle, then we must understand that suppose this is a skeletal muscle, and that you have studied that these are what T-tubules. These are what T-tubules, right? Now a very important point with respect to smooth muscle is this, that smooth muscles lack T-tubules. Smooth muscles do not have any T-tubules. Smooth muscles lack T-tubules. First understand this very basic point. Smooth muscles lack T-tubules. Therefore no invaginations, right? Now actually what happens is these small invaginations are actually present in the smooth muscle. Small invaginations are actually present at certain distances, but they are not as deep as T-tubules. Okay, they are not as deep as what? T-tubules. Now listen with all your attention. So these are what? Tell me, what are these invaginations? Now, what are these invaginations? It should not be big, that much deep. So like this. Okay, like this. Like this. Okay. Okay. So these are what? These are caveoli. These are what? They are known by the name of caveoli. Okay, they are known by the name of caveoli. Caveoli. Okay, caveoli. Okay, they are known by the name of caveoli. Okay, so these are your caveoli. Okay, now a very important point here we need to understand is this, that these caveoli are actually only some invaginations. If you think that these caveoli are actually some invaginations, then you are actually wrong. Why? Because these caveoli have specialized, now listen with all your attention, these caveoles have specialized voltage gated calcium channel in them, inside them, okay. These caveoli have specialized voltage gated calcium channel in them. Voltage gated what? Voltage gated sodium channel, voltage gated what? Calcium channel in them, okay. They have voltage gated calcium channel in them. These are what? These are voltage gated calcium channel. We'll talk about these, we will talk about the role of these calcium channels, so we will be talking about contraction, okay. Now for the time being, you must understand and you must believe that I am actually educated and I have little more activation in the cerebral cortex than you people, therefore you must believe my words. And these are what? These are what? These are voltage gated calcium channels. Clear? These are voltage gated calcium channels, okay. Clear? Now a very important point is this, that for the time being we will have to sacrifice our nucleus, okay. Now a very important point we need to understand is this, that suppose this is a smooth muscle cell, okay. Suppose this is a what? Smooth muscle cell. Now a very important point here we need to understand right now is this, that this smooth muscle actually have intrinsic and extensive cytoskeletal structures, okay. Now if I want to draw these cytoskeletal structures, you will not understand because I will be drawing other things as well. Now for that I have already prepared one model of this cell, okay. This is your model. Clear? This is your model. Now listen, don't worry, you don't have these much big cells in your stomach, okay. Your stomach actually is smaller than this. Now listen, this, consider this border as your cell membrane, okay. Now listen with all your attention. Suppose this is your border, or this is your cell membrane, okay. Those who are watching the video, they must also understand, okay. Now if this is the border of a cell, then what are these nets? You probably don't understand, you always read books. Now listen with all your attention. These nets, somebody was asking yesterday when we will be starting cell biology. Yes, we will start obviously, but listen, these nets are nothing but what are these nets? These are nothing but cytoskeletal elements. This is cytoskeleton. These nets actually, now listen with all your attention, these nets actually give shape to this cell membrane, right? And as a whole the cell, right? That means these nets are your what? These nets are your what? These nets are your cell cytoskeletal elements, and this border is your what? This border is your cell membrane. Now listen with all your attention. You are seeing some wires and some resistors, okay. You know what is a resistor and this is capacitor, okay. Now listen, very important point we need to understand here is this, that means I cannot draw these intrinsic structures here, okay. I cannot draw these intrinsic structures here, but I can make you people understand that what is happening here. Now listen, so you must believe me that suppose I have already drawn, okay, suppose I have already drawn the cytoskeletal network of this cell. Now listen, smooth muscle cytoskeleton is made up of mainly two protein. Now listen with all your attention. Smooth muscle cytoskeleton is actually Smooth muscle cytoskeleton is actually made up of two main proteins. Number one, desmin. Number two, vimentin, V-E-M-I-N-T-I-N, D-E-S-M-I-N, desmin and vimentin. That means the cytoskeleton means what? These elements, this meshwork, this meshwork has actually two proteins in them. That is what? Vimentin and desmin, right? Now listen, both vimentin and desmin are actually intermediate elements. They are actually what? Yes? Please say loud and clear. They are what? Yes, intermediate elements. Okay? They are intermediate elements. Clear? They are intermediate elements of cytoskeleton. Okay? Now listen with all your attention. Very, very important. We are going towards a very, very important, topic. Now listen. Now you understand now that what is the main difference between, clear? Now, what am I drawing, right? You are curious to know what am I drawing and what are these, right? What are these, things? Now, these are very, very important parts of smooth muscles. Now, listen. So, this is a smooth muscle cell. Now, listen with all your attention. Now, listen. Very important point we need to understand here is this, that, now listen. If I okay, now listen with all your attention. First understand this. What are these things which are present inside the cytoplasm of the cell? What are these things now? These are known by the name of dense bodies. Dense bodies, okay? These are known by the name of dense bodies. So this is one dense body. This is another dense body. This is another dense body. This is another dense body. Clear? This is another dense body. Now, a very important point. Very important point. So, this is another dense body here. Okay? This is another dense body. Now, listen with all your attention. Dense bodies are actually, anchors. Now, listen with all your attention. Dense bodies are actually the anchors of actin. Now, listen. Dense bodies are actually the anchors of actin and they are made by alpha actinin. They are made by alpha actinin. Dense bodies are, anchors of, they are anchors of, actin. Okay. Now listen. Where is actin? Right? Where is actin? Now listen. Here is your actin. These are your actin molecules. Okay. Here is your actin molecules. Okay. Here is your actin molecules. Clear? Now listen. Here is your actin molecules. Okay. Here is again your actin molecules. Okay. Here is again your actin molecules. Here is again your actin molecules. Now, a very important point here we need to understand is this. That means the dense bodies are actually, the dense bodies are actually the, starting point of every actin molecules, right? Right. Now, a very important point here is this. You need to understand is this, that they are actually the starting point, right? But what? They are what? These filament like structures are what? They are actin. Now my question is, where is myosin? Where is myosin? Now listen with all your attention. Myosins are actually present in between these actin. Why I am drawing like this, because they represents M, myosin. Clear? Myosins are actually present in between them. Okay. Clear? Clear? Myosins are present in between them. Okay. Here we have got another space. Put M everywhere. Now listen. Now, if I tell you that this is actually the, this is actually is the structure of, smooth muscle, then I will be fundamentally wrong. Why? Because if you just think for once, how will this muscle contract? How will this muscle contract? Because now listen, this is actin, these blue strands are what? Actin molecules, and these M's are what? Myosin molecules. These M's are what? Myosin molecules. Now listen with all your attention. If actin and myosin is interacting and they are coming together, then the whole muscle will not shorten. Why? Because these dense bodies are not connected to the, plasma membrane of the what? Plasma membrane of muscle. That means the peripheral dense bodies, that means the last dense bodies should be connected. Now listen with all your attention. The peripheral dense bodies, listen with all your attention. The peripheral dense bodies should be connected with these structures, these structures which I have already drawn, with these structures. Sorry, not with, caveoli, with these structures. Okay. Clear? Clear? Am I clear to everyone? Okay. Claro? Now listen. Now listen with all your attention. Now, if these actin and myosin are interacting with each other and they are contracting, that means due to the contraction here, this point will come and this point will also contract. That means this whole muscle is now connected. If I rub this green, strands, what will happen? What will happen? Then this whole muscle will never contract, right? Clear? Now, don't you tell me that if these green things are not there, that means it is single unit, and if it is there, it is multi-unit. No, it is not like that. You are proposing something like that, which is horrible. Okay, that means you don't know anything. It's okay. Sometimes it's better to say that your enemy lives within. Okay. Now listen. Okay, we are going too much, philosophical. Okay, here. So this is actually what? these ultimate ends are called dense plaques. Dense plaques, okay, PLAQUE, dense plaques. Clear? They are called dense plaques, okay. They are called dense plaques. Clear? That means these are what? These are dense plaques. These are dense plaques, okay. This is also dense plaques, okay. Now that means dense bodies are connected to the peripheral dense plaques by special actins, and therefore they are forming a, you know what, contractile unit. They are now forming a contractile unit. Clear? They are now forming a contractile unit. Am I clear to everyone? Okay. Now we were actually discussing what? Structure of smooth muscle. Now listen with all your attention. A very important point is this. Actually I have done one mistake, that is it will be called caveolae, okay. Sorry for my mistake, okay. It is called caveolae, okay. So this is your, you know, voltage grade calcium channels, okay. Now I will be rubbing this board and I will try to, you know, place two smooth, you know, muscles at a time, that is skeletal muscle and smooth muscle, and try to, you know, understand the structural electron microscope configuration. So suppose this is your, you know, smooth muscle, okay. Suppose this is your smooth muscle. Clear? Okay. What are these? Caveolae, right? These are your caveolae, right? These are your, yes please, caveolae. This is your caveolae, right? This is your caveolae. This is your caveolae. It will not go deeper, right? This is your caveolae. Now, sorry, it went very much deeper. It looks like T tubules, okay? Caveolae, okay? Clear? Caveolae. Claro? Now listen. Strange shapes. You're laughing. You understood what do you mean by this? Okay. Now, on the other hand, let's draw the structure of, you know, skeletal muscle. Okay. Now, suppose this is your, you know, skeletal muscle. It looks more like paramecium. Okay. Now, these are what? These are T tubules, right? These are what? T tubules. Okay. So the analogous structure of T tubules in smooth muscle is what? Caveolae, right? Or caveolae. Okay. Here, caveolae. So this is, you know, smooth muscle. You know, your skeletal muscle. Okay. Now these are your what? T tubules. These are your caveolae. Now listen. You know that suppose this is what? This is your what? These are your what? Dense plaques? No. These are dense bodies, right? Dense plaques are present in the periphery, but these are present inside the what? Inside the, okay. These are what? Dense bodies. Now, what are these things? What are these things? These are dense plaques. These are dense plaques. Okay. Dense plaques. Okay. Now listen. This is what? This is, you know, dense bodies, and these are what? Actin molecules, and in between them We have got, okay, in between them we have got what? In between we have got myosin, right? M, okay. So this is what? This is what? Myosin, okay. Clear? Now, okay, here we have got what? Actin molecules, right? Dense bodies are nothing but what? Dense bodies are nothing but, you know, anchors of actin molecules, and dense bodies itself are made up of alpha actinin, and in between them we have got what? The same, that is myosin, okay. Now we will be attaching, okay, let us draw with this color, okay, attaching this, you know, with what? This is called actin, and this is what? Dense bodies, right? Now listen, every dense body is connected to, you know what? Not all dense bodies, but peripheral dense bodies are connected to what? You know, dense plaques, right? Therefore a functional, actually, you know, unit is produced which can actually, you know, contract together, right? Which can actually contract together, right? Now listen with all your attention. Have you noticed one thing? Now listen. Now if this is your, you know, skeletal muscle, then what we will get? This is your, you know, this is what? This is your, you know, this is what? This is your actin. This is no, this is actin. Now listen. These are myosin, right? Clear? And this is what? Again actin. Now listen with all your attention. So these, you know, units are repetitive in your, you know what? Skeletal muscle. We have already seen that, right? Now a very important point here we need to understand is this, that this is what? Myosin, 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 right? This is myosin, 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 right? This is also myosin. Now listen with all your attention. A very important point here we need to understand is this, that, you know, what is the use of this line, you know already. This is what? This is what? Now listen, it looks like Z, like, right? That's why it is Z line. Okay, now listen. In skeletal muscles, Z lines are actin anchors, clear? But in what? In smooth muscles, dense bodies are actin what? Anchors, right? Dense bodies are actin anchors, clear? That means we have now learned what is the role of Z line and what is its similar, you know, counterpart in smooth muscle, right? Clear? Am I clear to everyone? Great. Okay. So this brings, you know, to the, you know, electron microscopic structure of smooth muscle. Okay. Now we will be talking about the second part, that is the contraction of smooth muscle. Okay. Before we start this, you know, second point in our lecture, that is the smooth muscle contraction, we must understand that we will actually be dividing the smooth muscle contraction topic into 3 different parts. Okay, dividing this whole topic into 3 different parts. Okay, 3 different topic, this topic into 3 different parts. Number A is entry of calcium, Number B is, you know, the role of calcium in muscle contraction, and C is relaxation of the muscle. Clear? C is relaxation of the muscle. So at first we will start with entry of calcium into the muscle. Clear? Okay. Now listen with all your attention. Okay. Okay. So now we are going to deal with entry of calcium into the smooth muscle cell. Okay. It is entry of calcium into smooth muscle cell. Now listen. We'll start with, suppose this is your smooth muscle membrane. Okay. This is your sarcolemma. Okay. Now we will be drawing this is your what this is caveoli, right? Okay. So we are going to draw what? This is nothing but what? This is nothing but your lipid bilayer, right? Now listen. I am drawing in a haphazard manner because we need to save the time as well, right? Okay. So this is suppose your what? This is suppose your cell membrane or sarcolemma. Okay. So this is your sarcolemma. Now listen with all your attention possible. The first way how calcium enters into sarcolemma is this, that now listen, is that there is one channel in the caveoli. There is one channel in the caveoli. Okay. Now listen. Any small fluctuations in the voltage of this cell causes this L type. This is a what? L type voltage gated calcium channel. Okay. Now any small fluctuation in voltage causes calcium to enter from extracellular space. Okay. Causes calcium to enter from extracellular space. Okay. Now very, very important point here is this is the first way by which calcium enters into smooth muscle cells. Okay. Now second thing is this, which is the most important thing, is this, that we will have to draw a special type of special area of we will have to make special area of cell membrane. Now listen, what is this area called? Now listen. Now if you compare this area, this specialized area, with other areas of cell membrane, what you will get is this, that here the phospholipids are attached in a very much what, sticky manner, right? they are attached together in a sticky manner. This area is called lipid raft. This area is called lipid raft, right? This area is called lipid raft. Now listen, what happens is this, in this lipid raft, actually what happens is this, in spaces of lipid rafts, G protein coupled receptors are actually present. Clear? G protein coupled receptors are actually present here. Clear? GPCRs or G protein coupled receptors are actually present. Now listen, these G protein coupled receptors are actually coupled to GQ. Clear? They are coupled to what? GQ. Okay? Now listen. Now listen with all your attention. So, when ligand or ligand, whatever it is, ligand binds with this, G protein coupled receptor, then GQ gets excited and it releases alpha Q, right? Now listen, what this alpha Q will do is actually it will, activate what? Phospholipase C beta, right? It will activate what? Phospholipase C beta, right? Now phospholipase C beta acts on, some special phospholipid, which are called what? Phosphatidyl inositol diphosphate or PIP2. They act on what? They act on specialized, phospholipids called PIP2, right? Now, when phospholipase beta acts on PIP2, PIP2 is converted into DAG, diacylglycerol, and IP3, which is inositol triphosphate. Now, suppose this is your inositol triphosphate, okay, IP3. Now listen with all your attention. What happens is this, we will have to draw here now what? You know that? We'll have to now draw, what is this? Sarcoplasmic reticulum, right? Now again we'll have to draw the, basic structure. Okay. Okay, so this is your sarcoplasmic reticular membrane. Okay, for the time being, understand that this is your membrane. Okay. Now, a very important part here we need to understand is this, that there is one channel which is IP3 gated calcium channel. Now in cells these are IP3 gated calcium channels. Now listen, this channel is closed when there is no IP3. That means here is the IP3 receptor. Okay, clear? Now when IP3 will sit here, when IP3 will actually sit here, clear? When IP3 will sit here, then this channel will open. Then this channel will open. This is a ligand gated channel, and that calcium concentration inside the sarcoplasmic reticulum is high, right? And therefore calcium will now flow out of the out of the channel, and it will come into the cytosol, right? This is actually the cytosol, right? Now, so it's very important that we have already discussed there are actually three mechanisms, but out of the three mechanisms we have already discussed two mechanisms, right? Now listen with all your attention possible. When calcium is moving in, now listen with all your attention. When calcium actually, due to some potential differences, when this calcium was entering the cell, then what happens is this: when this calcium was entering the cell, there was a calcium insurgency, right? Now, due to ligand also, and IP3 gated sodium channels may open, and calcium will come into cytosol, and when these calcium and these calciums are now present in the cytosol, then what will happen is this: a simple thing called calcium induced calcium release will occur. Calcium induced calcium release will occur, and this means that specialized calcium release channels will now be open, and they will release calcium from sarcoplasmic reticulum. Now listen with all your attention. You know what is the name of this channel? You know what is the name of this channel? You already know this. The name of this channel is ryanodine calcium release channel. Okay, this is ryanodine calcium release channel. Clear? This is ryanodine calcium release channel. Now listen with all your attention. When from these three sources, that is this and this, calcium is coming out, then it is called, now listen, the first two calcium entry into the cytosol is actually not calcium induced calcium release, but due to these two, the third entry from ryanodine calcium release channel is actually calcium induced calcium release. Now listen with, due to these three calcium due to these three things, calcium, due to these three events, calcium concentration in the cell increases, and this incidence is called calcium spike. Now listen with all your attention. This incidence is called calcium spike. Clear? This incidence is called calcium spike. Now listen with all your attention. What happens now is very, very interesting. Now listen. What happens is this: that means whenever calcium spikes occur, that means that all the channels through which calcium can enter the cytosol are actually open, whether it is present in the cell membrane or plasma membrane, or whether it is present in the sarcolemma or sarcotubular membrane. Okay. Now a very important point here we need to understand is this, that after a certain time, what happens is this: after certain times, specialized channel opens, and this is called potassium channels. These are what? Potassium channels. And when these potassium channels open due to voltage, they are also voltage gated sodium channel, and when this potassium channel open due to these disturbances in calcium spikes, they will start releasing potassium, and this cell will what? Cell will eventually go into what? Inactivation. That means calcium spike is actually calcium spike is the highest point or highest stage in smooth muscle contraction. Okay, but when this calcium spike results in the opening of potassium channel as well as chloride channels, okay, they also help in opening voltage gated chloride channels. And when these two channels are open, then inactivation of that particular smooth muscle occurs. Now, a very important point here we need to understand is this. Suppose these two channels are now closed, but in the intermediate stage of smooth muscle contraction, what is happening now is this, that calcium is moving out of the sarcoplasmic reticulum, right? Now, sarcoplasmic reticulum also needs to check the calcium released into the cytosol. Therefore, it should have a concentration of cytoplasm. It should have a minimum concentration of calcium in them. Now, listen with all your attention. There is a protein. Now, listen. There is a protein by the name of STIM. S-T-I-M. There is a protein by the name of STIM protein. Okay. Now listen with all your attention possible on this earth. A protein by the name of STIM protein actually is present in the sarcolemma. Clear? Sorry, in the sarcoplasmic reticular membrane. Okay. In the sarcoplasmic reticular membrane. Now listen with all your attention. Every STIM protein is actually a monomer, okay? But now listen, they have a calcium binding domain. This is a calcium binding domain, and to this calcium binds, okay? Calcium binds, okay? Now listen, when this calcium are moving out, then what happens is this, these STIM proteins now loses their calcium. They will now lose their calcium, and when they will lose their calcium, a very important thing will happen. You know what will happen? When they will lose their calcium, what will happen is this, they will not be monomers anymore. They will come together. Okay. They will come together and they will form, what? Tetramers. They will come together and they will form tetramers. Okay. They will come together and they will form tetramers. Okay. Clear? Okay. Now, very, very important point. So, STIM proteins, when they have lost their calcium from their calcium binding domain, they will form tetramers. Now, listen with all your attention. Every STIM protein, every STIM protein has two domains. Number one, which is associated with, what? Number one, which is associated with, sarcoplasmic reticular membrane, right? Another one is actually sticking out of the sarcoplasmic reticular, membrane, okay? Another one is sticking out of the sarcoplasmic reticular membrane. Now listen, when they become tetramers, they will activate a channel, calcium channel. They will activate a calcium channel here, okay? They will activate a calcium channel here. Now listen, these calcium channels are actually, now listen with all your attention. These calcium channels are actually, now listen, they will give, positive responses to what? These calcium channels. And now what will happen is this, when STIM proteins will form, tetramers, then what will happen is this, these extracellular domain of STIM proteins will actually activate this channel, and this channel is known by the name of ORAI calcium channel, ORAI calcium channels, okay? ORAI calcium channels. Clear? These are called ORAI calcium channels. Clear? Now, a very important part here we need to understand is this, that actually ORAI channels are actually, are also tetramers. That means they have what? They are also tetramers. That means they have 4 subunit. But for our 2D board, we can actually show the 2D figure. Actually, it is made up of, we have actually shown these figure. Okay, we have actually shown these figure. Okay, we have shown this figure, but actually it is made up of, suppose this is one subunit, this is another subunit of this channel. So there are actually 4 subunits. Okay, and when these 4 subunits come together, they form a what? They form a what? Channel. Now a very important part here is this, that these ori channels are very important. Why? Now listen, in lymphocytes, what is lymphocyte, right? In lymphocytes they have a important role to play. We'll talk about that later. But right now we need to understand is this, that when this calcium is moving in, now listen with all your attention. When this calcium is moving in, now listen, when this calcium is moving in, now you need to understand something very important. Okay, when this calcium is moving in, this calcium will enter, now listen, this calcium will now enter this calcium will now enter sarcoplasmic reticulum, okay, to replenish these calciums. Okay, because sarcoplasmic reticulum is very fast losing its calcium concentration, therefore calcium enters. Now a very important point you need to understand is this, due to the entry of this calcium, H plus will move out. So it's actually your what? Calcium H plus exchanger. It is calcium H plus exchanger. Okay, this protein is actually Ca plus to H plus exchanger. Clear? This is actually an exchanger protein. Okay, clear? Okay, so this is the entry of calcium into the cells. Okay, now listen, when this calcium will entry into the sarcoplasmic reticulum, they will bind with these what? Calcium binding domain of stem proteins, and therefore they will again become monomers. Okay, and they will never activate these channels again, ori channels again. Okay, clear?